Well, good afternoon. Welcome to Office Hours. We have a wonderful presentation today lined up by Dr. Rahul Gupta, who's going to introduce a method of quantifying mitochondrial DNA copy number at scale with the whole genome sequencing data with all of us and application to the UK Biobank. But before we get started, I do have a few announcements to go over before I pass the mic on over. So in case you missed our last Friday office hours, it is now posted to the User Support Hub under the video section. So the All of Us Digital Health Technology team presented an overview of the newly released Fitbit device data table, which is now available in the Researcher Workbench. So you can scan the QR code here if you want to learn more and get access to that video. Also, if you have questions about accessing the device table in the Researcher Workbench, please email us at support@researchallofus.org. I do also want to highlight a few events that are coming up around the Data and Research Center. First, our next drop-in office hours will be this coming Tuesday on the 14th at 1 p.m. Central Time. You all should have received an email to your institutional email address affiliated with your Workbench account with the link to the Zoom. If you need the Zoom link for Tuesday's drop-in office hours, please email us at support@researchallofus.org. Also, Baylor College of Medicine will be hosting their Evenings with Genetics next week on Wednesday at 1 p.m. Central Time. It's very office hours style, so if you have questions, feel free to join them. And then later on this month, there are two events that I would like to highlight on the 24th. The first one is Friday at 8:30 on 5/24. Our Ignite team is hosting an All of Us Researcher Workbench Friday morning coffee session, but that session is going to highlight grants and how to utilize grants, grant access, and how to prepare for grant writing, called Grants 101. So if you have questions related to grants, please join them. Also, that same day on the 24th, we'll be having an overview of using SAS Studio in the Researcher Workbench with one of our SAS experts. You can see more about our events on our event calendar at the User Support Hub. I do also want to note a few housekeeping things before we get started today. We please kindly ask that you put all chat messages into the general group chat. While myself and my DRC colleagues do appreciate you all directly messaging us, in order to efficiently communicate with you, we ask that you put your questions in the group chat. That also gives the opportunity of other researchers to learn from your question. We will have Q&A at the end of this presentation, but please keep all question and answers related to the presentation topic today. If you have specific general questions about Researcher Workbench usage or something going on with your account or a technical issue, please email us at support@researchallofus.org. Okay, I'm going to introduce our speaker today. Rahul Gupta is an MD, Ph.D. student at Harvard Medical School. He completed his Ph.D. in 2023, congratulations, and was co-advised by Vamsi Mutha and Ben Neal at the Broad Institute. For his PhD, Rahul used statistical genetics methods to evaluate evidence supporting the role of mitochondrial dysfunction in age-related disease and applied these methods to identifying mechanisms by which mitochondrial DNA variation can be influenced by the nuclear genome in humans. This later study, now published in Nature, is what today's focus is going to be in the talk. Prior to his graduate work, he earned his Bachelor's of Science in Engineering and Chemical Engineering from the University of Pennsylvania. Looking forward, Rahul aims to pursue clinical practice with research focused on using human genetics and novel molecular phenotypes to uncover mechanisms for individual variation in basic cellular metabolism, treatment response, and disease presentations. And with that being said, I will pass it over to you.
Thank you for the intro. Let me just share my slides. Are my slides up?
Yes, we can see them.
Perfect. So thanks for the intro. I'm really excited to get to present to this group and in this venue. All of Us is an incredibly important resource, not just for this study, but for so many other studies, and it's only getting better with more and more data. So really excited to get to share this work here. And as was mentioned, please feel free to ask questions and provide feedback on anything that I mentioned today. So with that being said, I will jump in. The topic of today's talk is nuclear genetic control of mtDNA homeostasis. revealed from more than 250,000 diverse human genomes. I'll start with some background on the mitochondrial genome and motivation for this work, and then we'll dive into the pipeline that was developed, as well as the results that we were able to glean from these data. So I think as many of you are familiar, humans carry two very different genomes. Our nuclear genome consists of two copies. It's linear, it's extremely large with over 3 billion bases, and carries approximately 20,000 protein coding genes. The mitochondrial genome is quite different in basically every way. It's circular, it's maternally transmitted, and it's very, very small. Each copy only has about 16,000 or so base pairs, and only codes for 13 protein coding genes. So extremely different in structure and function. And one of the most interesting features of the mitochondrial DNA is that it's a high copy genome. So unlike the nuclear genome, which is present in 2 copies per cell approximately, depending on which cell you look at, obviously, the mitochondrial DNA is present in any number of copies, and that number can differ from cell to cell, from tissue to tissue, and from person to person, making this a very dynamic molecule and making copy number a super interesting quantity, as we'll talk about. Mutations in the mitochondrial DNA are also just functionally different from what we think of in the nuclear genome. So first off, the mitochondrial DNA is almost exclusively maternally transmitted. So if a mutant mtDNA is present in the mother, all offspring will have that mutation, whereas if father carries a mutation in the mtDNA, that mutation will not be transmitted to offspring. Additionally, because mtDNA is present in many copies, the way that we think about heteroplasmy, rather the way that we think about mutations is different from how we think about in the nucleus. So in the nucleus, we think about mutations being present typically in 0, 1, or 2 copies. Whereas in the mitochondrial DNA, because we have a number of copies, we think about mutations as heteroplasmy, which is just the percentage of molecules carrying a mutation. So for example, this red allele is present in 100% of the molecules here. So it's at a homoplasmy or heteroplasmic fraction of 100%. And this blue allele is at a fraction of 10% because it's only present in 1 of 10 molecules. So you'll hear me say heteroplasmy throughout this presentation, and that is basically referring to the allele fraction within the sample that we're looking at. So I've touched upon 2 aspects of mitochondrial DNA phenotypes, the mitochondrial DNA copy number and mtDNA heteroplasmy. And these are the 2 quantities that we'll dive into during this presentation. Starting with the copy number, why is this something that we really care about? First off, across many different tissues, we know that copy number can vary widely. So in whole blood, for example, work that we've done in the past has shown that whole blood mtDNA copy number can be as low as on the order of 100 copies per cell, whereas we and others have seen that in other tissues like heart or brain, mtDNA can be present on the order of 500, 5,000, and even more molecules per cell. And so this is an extremely variable quantity across even just one person. The mechanisms that sort of govern that set point, some of them are known, but many are yet unknown. And clearly this has an impact on cellular energetics and other phenomena. We also know that across people, if you see these error bars on these box plots, across people, there is also quite a bit of variation. But the mechanisms governing that variation are also only just beginning to be understood. Further, the degree of variation across people may also be related to disease state. So previous work has shown that mtDNA copy number can decline in the context of cardiovascular disease and type 2 diabetes and other diseases as well, which suggests potentially a link to disease states that's worth investigating. And we'll talk about this throughout today's presentation. So copy number is highly variable and potentially related to aging and disease. What about heteroplasmy? I think heteroplasmy is one of the most fascinating quantities just because of how many different factors can govern how heteroplasmy can change from cell to cell. So for example, the mtDNA has a high mutation rate relative to the nuclear genome. Some people will estimate that at about 10x the mutation rate. And that indicates that there's just a like a higher risk for there to be somatic mutations in the mitochondrial genome. It's also been shown that mutation rate in the mtDNA actually varies as a function of strand and position, and that each of these mtDNA variants can actually have selective pressures for or against the variant that could differ from tissue to tissue and can determine the heteroplasmy that is seen. So for example, the MELOS variant was recently shown to exhibit tissue-specific selection in, for example, the T-cell lineage versus other lineages studied here. There's also a lot of drift that occurs due to bottleneck effects that occur in each generation. So the oocyte, which contains tons and tons of mtDNA, eventually has to form these primordial germ cells, which is essentially a huge drop-off in the mtDNA copy number, and that increases the variation in heteroplasmy across these cells. Further, there's also genetic drift, which occurs across tissues, which results in likely variation in heteroplasmy from tissue to tissue within one person. So these are all different mechanisms that have been either shown or hypothesized or both that can contribute to variation in the heteroplasmic fraction of a single variant in the mtDNA. But how all of these different things can interact at a population scale and contribute to variation in heteroplasmy in humans is not really super well understood. So the next thing to touch on is what molecules, what proteins contribute to mitochondrial DNA stability. So obviously you need proteins to replicate, transcribe, maintain, degrade the mitochondrial genome. Where do these come from? So the mitochondrial genome, as I mentioned, encodes only 13 proteins. And these proteins actually are all involved in the OXPHOS pathway, which is the main pathway through which aerobic respiration occurs. And the key point that I want to really, really emphasize here is that the vast majority of the mitochondrial proteome, over 1,100 proteins actually, come from the nuclear genome. So the mitochondrial proteome is quite extensive. Only 13 of these are from the mtDNA. The vast, vast majority come from the nuclear genome. And that includes every single protein that contributes to mitochondrial DNA maintenance and dynamics, including proteins responsible for nucleotide pool maintenance, mtDNA replication, mtDNA transcription, and degradation. So all of these mechanisms are governed by proteins, some of which are shown here, that are encoded by the nuclear genome. So summarizing all these key principles, we're interested in, as part of this study, to understand how it is that mtDNA abundance and heteroplasmy can differ across humans. We know that they differ across people. We also know they differ across tissues and cells. And importantly, all of the proteins influencing mitochondrial DNA replication, transcription, et cetera, are actually encoded by the nuclear genome. And so this led us to consider a study design that looked for mutations in the nuclear genome and saw how those mutations might affect aspects of mtDNA dynamics. And this is, what I'm describing here is a genome-wide association study. GWAS, which I'm sure many of us are familiar with here, have been widely used to great success in identifying genetic architecture associated with common disease as well as common phenotypes. Here is a recent GWAS for type 2 diabetes showing a number of nuclear genetic loci associated with type 2 diabetes risk. And this has, again, been super useful in understanding not just potential therapeutic targets, but also in understanding potential disease mechanism. But I think one of the most interesting applications of GWAS is to actually study extremely basic biological phenomena by identifying interesting intermediate phenotypes or molecular phenotypes. So one really cool example of this was recent work that looked at telomere length and measured telomere length across a large population of people. This study basically wanted to understand how the length of telomeres could differ across people. As I think many of us are probably familiar with, telomeres are these caps at the end of our chromosomes, which tend to decline in size as a function of cell division and aging, and tend to be longer in the malignant cancer state. And so what authors did was measure telomere length and perform a GWAS. They found a very extensive nuclear genetic architecture associated with telomere length, and using this, we're actually able to reconstitute the basically the majority of the known pathway of telomere maintenance at a basic biochemical level. And so I think this is super cool. And this is sort of what we were hoping to achieve by quantifying a whole host of mitochondrial DNA related traits, basically pull out the basic mechanisms that govern mitochondrial DNA biology. And again, we're aided by the fact that so much of mtDNA machinery is encoded by the nuclear genome. We were reasonably well positioned to do this because at the time that we started the study, whole genome sequences had just been released from UK Biobank and all of us, and at that time had totaled 250K individuals. And so we built the tools to try to quantify mtDNA copy number, but also quantify carefully mtDNA mutations at very large scale. And since the publication of this paper, even more data has become available, which we've been very excited about continuing to pursue and extend these analyses. So what I'll now dive into is our pipeline for estimating copy number and mtDNA variants. I'll talk a little bit about how the pipeline works and how it can be used in the all of us data. Then we'll talk about mtDNA copy number and what we found from that analysis, and then we'll finish with mtDNA heteroplasmy. Starting with the pipeline, we wanted to try to address three major challenges in the analysis of mtDNA. The first is that variants exist in fractional quantities, which is unlike the nuclear genome. And so traditional tools that analyze nuclear genetic code could not be used, and we had to turn to reasonably well-validated tools that focus specifically on the mitochondrial genome. Secondly, we had to deal with these nuclear insertions of mitochondrial DNA origin, which tend to be a huge nuisance in this type of work, which I'll talk about briefly. And then third, the reference genome in the GRCH38 reference has a very particular haplogroup H2, which is a European ancestry haplogroup. And so individuals with other ancestry, mitochondrial ancestry, will tend to have many more deviations from the reference. And we believe that this could potentially impair read mapping to individuals in an ancestry-specific way. And so we want to try to address that as well. Speaking a little bit about these new mites, these are actually really cool. Basically, it seems like in sometime in the ancestral past, several regions of the mitochondrial DNA have broken off and inserted themselves into the nuclear genome. Here is an example in chromosome 1 with these pseudogenes that have been identified, which are likely from the mtDNA. And while cool, this actually results in a big problem in the context of variant calling, because you could imagine that there are nuclear DNA sequences which get sequenced, and then the reads get mapped to the mitochondrial genome incorrectly. And that can result in a situation where you have low heteroplasmy variants that get called as mitochondrial and thus are artifactual. So we wanted to try to minimize the risk of this confounding our results. The approach that we took briefly was basically to do two passes of variant calling. So the first pass was we took the mtDNA and we took known new mite regions, and we called high confidence homoplasmic and homozygous variants. And then we built a consensus sequence for each individual for the mtDNA and for these new mite regions. We remapped reads to these regions, and then we recalled variants now with respect to each individual's consensus sequence, and then we mapped everything back to a reference sequence, with the goal of minimizing read mismapping and maximizing the chances of us getting ideal mtDNA coverage and reducing the risk of this type of artifact. And we do see some gains with this type of approach. So here is mtDNA coverage which is generally uniform or expected to be uniform with whole genome sequencing. And we find that our pipeline, for example, produces much more uniform coverage in this individual than the vanilla pipeline. And these dips here can actually be directly mapped back to nuclear regions that show homology to these sites, and previously were sort of stealing reads from the mtDNA. This pipeline was written in a WDL format, and I pasted a QR code here pointing to our GitHub for this pipeline, which is available for use. This has been successfully deployed on Cromwell, on All of Us Workbench, as well as on Terra. And then there's another version of this pipeline, which is also posted here, which we've deployed successfully on DNA Nexus for UK Biobank data. These two pipelines are different, just to optimize them for the different platforms. So the single pipeline operates on single samples, one at a time, and it tends to be faster on Cromwell. The multi-pipeline analyzes multiple samples at once. It tends to be faster on DNA Nexus, which supports that type of analysis. And so, feel free to try to use these pipelines for analyzing mitochondrial DNA data or whole genome data on these types of platforms. Moving on to the results that we were able to come up with. Once we ran these pipelines across UK Biobank and All of Us, the first thing we wanted to look at was mtDNA copy number. And particularly in the UK Biobank, where we have all these phenotypes, we wanted to see what variables could correlate with mtDNA copy number and potentially impact our results. The first thing we looked at was blood cell type composition. So different blood cell types in the whole blood that was analyzed, we thought would have different mean MTCN. And so we wanted to make sure that we either addressed potential confounding due to differences in blood composition or verified that there was no confounding. And what we found was that several blood cell fractions, such as neutrophil percentage or platelet crit, actually correlated very significantly with mtDNA copy number. And these are, population scale correlations, but these are some pretty significant correlation coefficients. Once we corrected for blood cell composition, we found some pretty interesting other correlations as well, such as what looks like some seasonal component to whole blood MTCN, some correlation with when in the day you draw blood, fasting time, etc., assessment center even. So, these are not variables for which we have a good explanation for why there's variation even after you correct the blood composition. But I think it really highlights how sensitive MTCN can be to technical variables that are otherwise unanticipated in this type of analysis. So we built models to correct for blood composition, these technical variables, as well as other demographic variables. And we find that these models together can explain more than 25% of the variation in MTCN, which is a huge percentage. And most of that is actually these blood cell composition traits. These other things do explain some amount of variation, but not nearly as much. We then wanted to better understand the associations between MTCN and disease. So we correlated MTCN with a number of disease traits. Here I'm only presenting 5. In our paper, we show, I believe, 28 or 29 different diseases, like particularly common diseases that are age-related and associated with MTCN in the literature. And here we were able to totally replicate what's been seen, which is that there is a very significant decline in MTCN in the context of patients with diseases such as cardiovascular disease, stroke, type 2 diabetes, and others. However, once we corrected for blood composition, we found that basically all of these associations sort of vanished in our hands, and in some cases actually reverse directions, such as in the context of MI. And what we think is going on here is that people with these different comorbidities tend to also have like some sort of systemic inflammatory milieu in their blood, as evidenced by the fact that individuals with these diagnoses tend to have increased neutrophil counts and in some cases increased platelet counts, which is another acute phase reactant. And so we think that this is likely confounding these MTCN patients. We'll get into the GWAS at the end of this section, but just wanted to showcase that if you do the GWAS without correcting for blood composition, you'll end up getting a bunch of extra peaks, such as these CXCL2, HLA, CDK, HBS1L, gasdermin genes. And these are like inflammatory in flavor, and suggest that likely these genes are acting on MTCN via blood cell composition, and correcting for blood cell composition basically eliminates all of these genes. Just to drive this point home and sort of be as rigorous as possible, we wanted to understand how could MTCN and blood composition be related. I think my assumption going into this was that you could have some factors influencing blood composition, which would then change your bulk MTCN. But you can imagine another model in which somehow something could affect MTCN, and that could actually shift blood composition downstream of MTCN. And so to dig into this, we performed a bidirectional Mendelian randomization analysis in which we took genome-wide significant loci for MTCN, and we asked how that affected or correlated with effect sizes for neutrophil count, and vice versa, took genome-wide significant loci for neutrophil count and asked how that could impact MTCN. What we found was that neutrophil count associated variants were highly correlated to MTCN effect sizes, suggesting that you could modify genetically neutrophil count and then affect MTCN. But we found that variants that were found to associate with raw and adjusted MTCN actually did not really correlate with neutrophil count effect size, suggesting that if you modified MTCN by modifying some of these mitochondrial DNA related proteins, you would not really shift to neutrophil count. So that really supports this theory that likely there's some inflammation going on that's shifting neutrophil count that's then impacting MTCN in these scenarios. So all of this analysis that I've talked about so far has been using UK Biobank data. We also have an additional set of data from all of us, which we were really excited to use. However, we actually ended up not using these data in our paper, and that's because the all of us data for mtDNA copy number actually showed this sort of interesting bimodal distribution, which we believed is most likely some kind of a technical component here. This is too much of a MTCN difference to really make sense as like a true biological difference. Like this is almost 2x the mode here from the lower mode. And we tried looking at variables like sequencing center or assessment state, which we had access to, and that did not fully explain this difference. We don't know what this bimodality could be due to because we didn't really have access to additional variables. But some explanations could be, for example, if certain blood draw centers focused on buffy coat versus whole blood extraction, or if DNA extraction methods differed across different centers or different blood draw sites. But we do not have access to the additional like covariates that would allow us to understand what is driving this difference. And so because we were afraid of potentially adding in some additional confounding without appropriate corrections, we ended up not including MTCN analysis from all of us data. But it's something that we would love to revisit in the sort of current release, the current updated release of all of us. So from the UK Biobank, this is now in about 150,000 or 180,000 individuals. This is what our final corrected mtDNA copy number GWAS looked like. So these red genes are genes that are either mitocardia or mtDNA disease genes, and these like tick marks, etc., are annotations that correspond to whether there's a coding variant, whether it's fine mapped, confidence of fine mapping, etc. But what I really want to highlight is many of these genes are mitochondrial genes. They are genes that associate with mtDNA. DGUC, for example, is known. Mutations in DGUC can produce Mendelian disorders of mtDNA depletion. And so it's really cool to see that common variants in DGUC can also have subtle variation in MTCN. We were also excited to see that there's a number of other genes that we don't have obvious explanations for how they could be related to mtDNA copy number, which could highlight some potentially interesting novel mechanisms of mtDNA regulation. All right, so that was our work on mtDNA copy number. In the last section, I'll talk about our work on mtDNA heteroplasmy and how using the diverse genomes from the All of Us data set really helped us understand mechanisms of how some of these associations may have come about. So the first thing that we did was, once we had this call set of high confidence heteroplasmic variants across the UK Biobank and All of Us, we first looked at where these variants appeared to occur throughout the mtDNA. And heteroplasmic SNVs appeared to be somewhat uniformly distributed throughout most of the molecule, with what looks like kind of a bump near this control region, which is this non-coding region at the top of the molecule. This top area, the control region, is the largest non-coding region in the mtDNA. Most of the rest of the mtDNA is actually coding and is very dense compared to the nuclear genome. Indels showed a very different distribution, where much of the coding region actually had no detected indels at a sufficiently high heteroplasmy for us to find, except for maybe some peaks here, which may overlap with some of these non-coding regions actually between genes. But then in the control region, there's actually a huge preponderance of indels that we found, particularly in this D loop area here in the non-coding region, control region, as evidenced by these peaks up here. So what we think is going on here is maybe some kind of a negative selection phenomena where indels that crop up in the coding regions may just not be tenable, may be competed out by other mtDNA molecules and thus be selected against, whereas the same degree of pressure is not really present in this non-coding region. We wanted to then look into its relationship with age. And so we were thankful that All of Us data contains individuals that are like above the age of 70 and are quite old compared to individuals in the UK Biobank cohort, which tend to be younger on average. And this really gave us the resolution to see this previously observed increase in mtDNA variants as a function of age. And it was pretty interesting to see that this sort of distribution really sort of takes off after the age of 70, where it's pretty flat before the age of 70. Interestingly, this appeared to be specific to SNVs, where the indels that we found mostly were pretty flat in terms of their abundance throughout the age distribution. And this was unexpected. I think the SNV result we had anticipated based on the literature, but not the indel result. We sort of expected these to behave similarly. The fact that they look different implied that somehow either how they were transmitted or how they were generated was different. And so to better understand that, we looked at transmission of these variants using trios in the UK Biobank dataset. So these are heteroplasmy transmission plots in UK Biobank. I'll walk you through what this one looks like and what this one means. So each dot here is a heteroplasmic mutation that is present in either mother or offspring. So if we look up here, this point here corresponds to a variant present at 75% in mom and 50%-ish in offspring. And so the first thing you might notice is this like correlation of points here. So these are variants that are present in both mother and offspring, and the fact that there's a correlation implies true transmission of these variants from mother to offspring, because you can predict offspring's heteroplasmy based on mom's heteroplasmy. There's also these points on the X and Y axes. These are not transmitted because they're found only in one and not the other. So like here, this is a variant only in mom. Here's a variant only in offspring. So there's some variants that are not transmitted, some variants that are clearly transmitted. And based on the marginal distributions, you can see that most of the untransmitted variants are SNVs and most of the transmitted variants tend to be indels. If we go to the father-offspring correlation plot, there's not really a correlation between father and offspring, which is expected. And then the sibling-sibling correlation plot mirrors what we see in mom and offspring, and that makes sense because both siblings will inherit the same mtDNA as mom. So what this taught us was that SNVs, on average, tend to accrue with age, and they tend to not be transmitted. And so they're more likely to be somatic than the heteroplasmic indels, which actually are quite stable with age and are quantitatively transmitted from mom to offspring and match between siblings, and thus are more likely to be inherited. this also would explain this distribution here that we see. We repeated this analysis in Terra using 1,000 genome cell line data, and we actually found a similar phenomena where even though there's more noise here, we still see what looks like a correlation between mom and offspring, between indel heteroplasmies, but not SNV heteroplasmy, which are more likely to be exclusive. And then again, no father-offspring correlation. I just want to take a moment here because I think this is pretty cool. This implies that these heteroplasmies are reasonably stable, that you can go from a heteroplasmy in an embryo, make it through a bottleneck, go through primordial germ cells, oocytes, which then get fertilized into a new embryo, which then differentiate into like stem cells and blood cells, and through all these stages, still have correlations in heteroplasmy. And then the 1,000 genome cell lines, which are EBV transformed blood cells in Petri dish, actually end up also being reasonably stable in terms of their heteroplasmy. So I think it's a pretty impressive phenomenon, and it's cool to see these play out in our population level data. So going back to our distribution of heteroplasmies, here are our indels, which we mentioned were super frequent. What we were just talking about was the overall distribution, but for the last part of this presentation, we focused on the indel specifically, and particularly focused on this one indel here, which is the most common variant that we found in our data set. Now, this site is pretty interesting in itself. So this is position 302 on the mtDNA. And this site is actually in this really cool region, which forms this tertiary structure called the G quadruplex. And the structure is thought to be related to a mitochondrial transcription replication switch, where this RNA sequence actually gets terminated or has the option of getting terminated and then is used as an RNA primer for replication. And this site has all these poly C and poly G repeats. And this is where we're seeing our variation in the length of these repeats. This plot here is showing the population distribution of the variants at this site in the UK Biobank, where each column corresponds to an individual, and the colors represent the composition of variants at the site. So half these people have the reference mostly, but then the other half of these people actually have combinations of multiple different alternate variants. So like this person here has a large percentage of red allele, some amount of the blue allele, and some amount of the green allele at this very position. And these heteroplasmies are also maternally transmitted from mom to offspring and are rarely exclusive to each individual. If we look at the distribution of heteroplasmies across people who carry them, you can see that there's quite a bit of variation. And so similar to the UK Biobank copy number analysis, here we can ask a similar question. We can ask what nuclear genetic loci contributes to variation in heteroplasmy across the human population. So we performed a GWAS with these heteroplasmies as our trait of interest. And what we found was, I think, quite amazing, which is that there are nuclear genetic variants in genes that actually directly bind to the mtDNA that then can influence mtDNA heteroplasmy. Many of these genes like SSBP1, TFAM, TEFM, POLG, et cetera, are thought to bind directly to the mtDNA and contribute to its replication. So this is like a really cool direct trans interaction between the two genomes. The SSBP1 locus was particularly interesting, because of its function, but also because it's such a strong association. But we were a little surprised that we were unable to find map this locus. And when we looked a little bit closely at the locus, we actually found potentially concerning was that it was reasonably close to this region with homology to the mtDNA. So here's SSBP1, here's our lead SNP, and then here's the mtDNA region with homology, the nuclear DNA region with homology to the mtDNA. So we're worried maybe this could be confounded by a pneumite or some other sort of artifactual result. But I think we were really supported by our multi-ancestry analysis. So in all of us data, we were able to look at this association just within individuals assigned to the African ancestry group. And in this group, there's much lower LD between the area of interest and our potential region of homology. And so the fact that this association still exists offered support that this is likely a real association and is not driven by whatever is going on here. We're also supported by a rare variant analysis in which we looked at ultra rare missense and loss of function variants within the SSBP1 gene, and we found another association between SSBP1 and our heteroplasmy. And this association is not recapitulated when looking at just synonymous variation. And so this is also inconsistent with confounding from a new one. So we have a number of these other mutations that are common in the population. And so we performed repeated GWAS across all these other heteroplasmies. And this is a plot that basically collapses a number of GWAS into a small area. Each row is a Manhattan plot, and each dot, like the size of the dot, is the height of the bar in the Manhattan plot. And what we immediately saw was that there appears to be some really interesting heterogeneity, where some loci, like PNP, appears to be correlated with a number of mtDNA sites. Other loci appear to be specific to certain mtDNA positions, but not associated with other mtDNA positions. And so super interesting that we're beginning to see some degree of sort of specificity between how the nuclear genome could associate with different variants and different locations of the mtDNA. Using the all of us data, we were able to show that the effect sizes calculated across the two data sets are pretty similar. They're really well correlated with some degree of attenuation as expected with Winner's curse. And in fact, the UK Biobank meta-analysis replicated quite nicely with each individual all of us ancestry group, such as AFR, AMR, EAS, and EUR. So in the very last section, I just want to speculate a little bit on what we think is going on and how it is possible that you can have these nuclear genetic variants that then impact heteroplasmy in the mitochondrial DNA. So as I mentioned earlier, one of our sort of most interesting sites is this 302 location, which is responsible for switching between mtDNA transcription and mtDNA replication. And we find that there's this nice transacting control where like you can modify the sequences contributing to the very proteins that bind to the mtDNA and influence this exact position. We also found different alleles in the mtDNA position are correlated with mtDNA copy number. So the way that we're sort of piecing this together, at least at this site, is that it's possible that by modifying the nuclear genome and by modifying the proteins that then replicate the mtDNA, you're actually potentially shifting the preference of these proteins to replicate one of these mtDNA genotypes over another. And that's how, and particularly because this is a site of transcription replication switching, and that's how you can shift heteroplasmy subtly across the population by changing the nuclear DNA haplotype. So I will end there. Just some conclusions. mtDNA copy number varies across the population and is influenced by a very vast number of loci. And blood composition is a really important confounder. And in fact, copy number does not appear to be associated with many diseases once you correct for blood composition. Heteroplasmic SNVs appear to be more likely to be somatic and accumulate with age, while most heteroplasmic indels that we see tend to be maternally inherited and do not tend to be somatic. There's many nuclear loci that control heteroplasmy, and we think that at least some of these loci may act to be like a replication advantage type mechanism to influence mtDNA heteroplasmy. So I will end there. Thanks for listening, everyone. Happy to take questions, and just want to give a shout out to my advisors, Ben and Vamsi, without whom this project would not be possible, and also to UK Biobank and all of us for providing literally all of the data on which this project was based.